I'll introduce Yael. Yael is our immunoassay manager in the HIMC, and she has actually been with the lab for at least 5 or 6 years, I guess, working on Luminex and other immunoassay technologies. You're going to use this?
I think so. Okay. I'm going to try and put the...
Then let's plug you in. Anyway, so in that time, Yael has gotten a lot of experience with how to handle Luminex data, what the common pitfalls and problems are, and so she's going to tell us a bit about that and comparison of Luminex to another technology that we have in the lab, which is the mesoscale discovery platform. So, thanks, Yael.
Hello, everyone. So I will describe two current immunoassays that we run at the lab, the Luminex and the MSD. And just to reinforce, the interest in cytokines is obvious, being mediators of the immune response. And actually the challenge would be to detect the ones that are at the picogram level, at the low picogram level. So just a bit about the technology. So the Luminex is a multiplex bead-based ELISA where basically the sandwich ELISA is built on a bead. These beads are created with, are dyed with to create different, up to 100 different spectral addresses. Each bead will be able to detect one typical cytokine, and we have multiple of those in each well. All cytokines are measured simultaneously in one well. It's done in a microtiter 96 well plate. And basically, once you perform the ELISA, which is captured on that bead, you run that through a flow cytometer, which detects, first of all, the red laser will detect the bead ID, meaning what cytokine it is that is captured, and then the green laser will actually intercept and detect the amount of fluorescence that is bound to that bead, meaning the amount of cytokine bound to that bead. The mesoscale platform is an electrochemiluminescence platform. It's a little different because it's a pre-patterned array captured at the bottom of the plate, and it's limited up to 10 cytokines per well. So that's the limit of the number of cytokines you can measure. Whereas I did not mention before, but for the Luminex, you can basically go up to 100 different cytokines. So for the mesoscale platform, this pre-patterned array, again, you're building a sandwich immunoassay at the bottom of the well over there, and you will see that the signal is intensified using the chemical reaction there, and the reaction actually emits light, and the light is captured with a CCD camera. So the sector imager, which is the instrument, reads 4 wells at a time, sections of 4 wells at a time. It's very fast, and it completes a plate in minutes, whereas the Luminex is a little slower in terms of the capture of the data. So when we just compare the two platforms, each of them has a different output, meaning fluorescence for the Luminex, whereas it's light intensity for the MSD. Again, for both, the amount of sample that you use is 25 microliters regardless. The output is we have about 55 measurements that we're performing for the Luminex and up to 10, as I was mentioning. Those intensities or median MFI fluorescence can be converted later to picogram per mil, and I'll touch upon that a little later. So what type of samples do we run on this assay? The ones in blue are the most common ones run in the lab: serum, plasma, tissue culture supinatants, tissue homogenates, and others mentioned here like urine, cerebrospinal fluid, etc. One thing that I really want to mention here, because this is a good time for you to consider, don't mix plasma and serum. Remember that they're different. Remember that the processing of them is a little different, and be consistent. If you start an experiment with plasma, be consistent and continue with plasma. It's not interchangeable with serum. Same for serum. In terms of normalization, when you're looking at tissue homogenates and cell culture supinatants, remember that you need to normalize before you run. Meaning, for tissue culture supinatants, it's common to normalize to the number of input cells in a well if you're stimulating or any experiment that you're performing. Whereas for tissue homogenates, you need to measure the amount of protein, normalize to a common amount of protein, and then compare those samples. So I'm going to touch upon this more in detail in the demo, and you're welcome to come up with a lot of additional questions over there. All I'll say at this point is that we typically run replicates of 2 replicates per sample. Our CVs are pretty good, below 10% for the most part. Only about 10% would be above 20% CV, which is coefficient of variation. I need to make one apology. The next slides that you're going to have, that you're going to see in the presentation, are not like what you have in your handout, and you're welcome. I have those also at the bottom of my presentation if you want to ask questions, or in the demo. But they are going to be different because I've included a lot of additional analysis since last year. Again, we use, to go back to our quality control checks, we use beads, which are called SHX beads. They're made by a company called Radix Biosolutions. Again, will be touched upon in the demo more in detail. The other thing that we include in all of our runs for human and mouse are serum controls. So we have a human serum control mix, and we have a mouse serum control mix that we include in every plate. This is going to be very important in the next slides that you will see. So this is just to lay out the 51-plex, which is a polystyrene-based kit. And in red are the 4 cytokines that I'm going to go a little more in detail. So we measure median MFI fluorescence or intensity. And now the question is, how do we convert that into a comparable measurement to compare between samples, between plates, etc. So one very common way is to include a standard that is serially diluted. A standard would be a known amount of concentration of all the analytes that are included in that assay. Serially dilute those, create a graph that is depicted here, both for the MSD and for the Luminex, and convert the signal that is captured on the instrument into a picogram per mil equivalent. Now, note that towards the lower end of the curve, so there's 7 points of dilution, towards the lower end of the curve, it is a little more sigmoidal, or a little less linear, and that forms some difficulty in terms of quantitation. Okay, so before I go on to the next slide, I'm going to concentrate. I need to reinforce again, we use our human control. I'm going to concentrate on our human control, or what we call CONS2. It's the second control that we've been using in the lab, and it is included, as I mentioned, in all of our assays. One topic that is very important into determining what is best to report. Is it better to report the MFI, or the intensity, or the picogram per mil? To help us with this answer, I'm going to address the topic of matrix effect, specifically complex matrix effect, as seen in plasma and serum. Those are more complex because they contain a lot of different components and hence more of a challenge in terms of quantitation. So in this spike-in experiment, what we did, we used the standard, one of the higher standards, standard number 6, spiked that into serum and measured and compared the detection of that S6 in the serum. So as you can see, the blue line is the level of all 51 cytokines, and you don't see all of them laid out here, but they're individually labeled like every 3 or 4 cytokines. And you can see some of them, there's a very big difference, very big gap between the detection of the S6 versus its detection, the same S6, as detected in serum number 2, our control serum, and serum number 3, which is a different control serum. So what does this effect tell us?
I just realized that this is a little confusing because S2 means serum number 2.
Yes, it's serum number 2.
Serum number 3, but
Yeah.
S6 means standard.
Yes, sorry. Yes, I have to. Yes. So just to be clear, Holden helped me clarify that one. S6 means standard number 6 spiked into serum number 2. And serum number 6 spiked into serum number 3. Oh, yeah, I didn't even look at that. I see it always as a given already. And then S6 with nothing, the blue, is just that standard as detected in the assay. So you can see that the matrix...
What does the standard contain? It's a mix of all the cytokines?
Yes, the standard contains a mix of all cytokines. And you can see that the level detected in that standard number 6 is much higher than what we're seeing when we put that standard into serum. So what is this telling us? And basically what it's saying is that we're underestimating the amount that's in a serum sample in this assay. That's the first thing that it's letting us know. The other take-home message would be that I haven't shown here, but with dilution, you actually improve this effect. So you reduce the amount of inhibitory effect. What is the reason that we see this inhibitory effect? This is just to show you that this is the same logged data with the baseline of the buffer. In purple is the baseline of the buffer, just buffer. The interesting part is that for the mesoscale platform, this effect is not seen. One thing to remember is that the mesoscale has each of the capture antibodies attached separately, physically located in different locations in the plate. So that may explain some of this effect that is not seen in the mesoscale platform. Where does it come from? Well, it's a mixture of inhibitory antibodies or soluble receptors, heterophile proteins that may interact and disturb and prevent us from detecting the true amount that's in the serum. And again, I think the take-home message is that where possible, in order to avoid this effect, the best is to dilute your sample. The other issue is to consider the fact that you're underestimating when you're converting the MFI into picogram per mil. It's somewhat underestimating the amount of protein in the sample.
One comment. I mean, if you're looking at serum, because, like, for example, if we're talking about immune responses, there might be these inhibitory molecules that work even there. So that might not necessarily be a bad thing. I mean, yes, I mean, if from a chemical standpoint, if we want to take the absolute level of the cytokine,
exactly.
That's not what we want, right? We want to see what the biological...
That's what I'm trying to persuade you.
Okay. You already have things.
So I'll tell you, the bottom line of it is that we really recommend, while it's very important, historically people were measuring ELISA, single single ELISAs and trying to come up with a quantitative measurement of that protein individually. Now we're moving to a multiplex era where we're trying to detect a pattern of cytokines, not only a single cytokine. So we're actually using kind of a relative pattern recognition to look at our data. We're not so interested in the exact quantification of a certain cytokine or biomarker. We're more interested in the relationships between them. And so we recommend, actually, in order to avoid a lot of other issues, to use the raw data, the median fluorescence intensity, and to use the intensity in MSD. And I hope it'll be more clear in the next few slides. So, okay, we dealt with the matrix effect. And now I just want to show you reproducibility. Again, using our control sample, our human control sample, this is across 20 different plates of the flu, a big flu study that we've been conducting for a couple of years in the lab. And you can see that they really overlap each other. This is true for the polystyrene assay, 20 plates here. It's true also for the magnetic cytokine assay. This is a different study run. It's a 41-plex, it's a 42-plex actually assay, where we, again, included our control. All of this is only looking, no samples here, only the control. That same control is run over and over again. This is in a magnetic 41-plex assay.
What is your axis? This is just fluorescent intensity? Or
So on the axes, this is only fluorescence intensity, exactly. I'm not presenting the picogram per mil. True for here and here.
So what is your cutoff of positive signal?
I'll get to that in a minute. So this is just another way to look at that. If we look at our control sample run multiple times, now I'm just flipping the axes in terms of the presentation. You can see these are the different cytokines all presented here, pretty reproducible. Just to show you, for MSD we ran on those same master plates. We use a master plate and apply those samples both in, test them both in Luminex and in the MSD, a 4-plex MSD. So this is again the equivalent of the 4-plex MSD for all these controls repeated in the same 20 plates. So then, why do I say that we like the median fluorescence intensity? The reason that I say that is on the top part, you can see the reproducible MFI presentation of this control sample. This is in another study. When you convert it into picogram per mil, what you basically see here is that, I don't know if you can see very well, but some cytokines become more spread apart. Remember, it's the same sample. The MFI is very reproducible. It's actually less than 20%, whereas the translation into picograms per mil creates a variability that is not there. So not only is it not standardizing, but it's actually creating more variability. I think it's can be more appreciated in this slide, where we look at the coefficient of variation. So you see the coefficient of variation for the most part on the left column is of the MFI, basically less than 20%. When we convert that into picogram per mil, what you see is it's conditionally formatted to show above 35. Some of them are even 100 and more. But you see that it's converting it in a way that creates more variability between the picogram measurements. And the reason for this, actually, a very small difference, as you can see, as you approach the lower end here, a very small change in MFI will lead to a much greater change in picogram per mil. So you can very easily move with a very small change of MFI to a change of double or more for when you translate that into concentration. This is only one example. Obviously, each cytokine behaves very differently. You have to look at each cytokine individually, and you'll appreciate that each of them will behave differently close to the lower end of the curve and the limit of detection. So then you can ask, how reproducible are our standards? Are our standards reproducible? And what I'm depicting here only 4 of the cytokines just to make it a little more simple. And what you're seeing is repetitions of that standard curve during different days. This is a 7-point standard curve, and this is what I want to stress here. So remember at the beginning I told you that we take a known concentration of those cytokines that are our standards, and we serially dilute them. A 7-point curve, and this is what we get. And you can see pretty much here, this is for MSD. You can see that there's somewhat little more variable when you approach the lower standards. This is more noticeable when we look at the Luminex, where the spread out between S3 and S1 is much more noticeable for IL-1 beta, for IL-6, somewhat for TNF alpha, and also for IL-8. It's true for the magnetic standards as well. Again, repetitions. Now, to sum it up, and this is only one example. I can show you multiple examples like these where we look at the CVs of the standard curves, put it in numbers. And what you see is that basically the first half, standard 1 through 3, is much more red. It's conditionally formatted so that red is above 30% CV. White is between 20 and 30% CV. Green is below 20% CV. You can see that basically the lower end of the curve is mostly red and white, whereas the upper end of the curve usually is a little more linear, therefore, and performing better and more reproducible over time. One thing to note here, and I'll elaborate on that during the demo, at the end there you can see there's checks 1, 2, 3, and 4, all are green. So that is a measure for our work, for our control. All wells are okay, performance is within accepted range, and is less than 15% for most parts, even lower.
Do you want to say what the checks are?
I can say briefly now just what they are. So checks 1 checks the performance of the instrument. Checks 2 checks the addition of strep-evident phycoerythrin, which is the last step of the ELISA assay. And checks 3 checks the addition of the detector antibody. So anything not within a 30% range would be not acceptable, meaning the well could be disqualified as to not receiving enough detector antibody, not receiving enough SAPE, etc. The last checks 4 antibody checks for non-specific interactions. Obviously, that is going to be higher for serum and plasma, and you'll see some examples in the demo. In general, the CV there is acceptable as higher because sometimes the numbers are very low, 1, 2, 3, 5, 6. So any variability between 4 and 6 is going to indicate a higher variability, but it's not really significant.
So in case people didn't get it, so those checks beads are 4 different beads that are spiked into every sample.
Every well.
Every well, yeah. In addition to the beads that are outside. And so they check for the specific things that they check.
So they're an independent measure for the performance of that well in addition to the sample. The only thing that we cannot guarantee, it leaves the sample itself. So if you enter inadequate amounts of sample or you forgot to add a sample, you'll see that in the CVs of those replicates, but you won't see anything in the checks data if the well ran well. So what I wanted to make now is actually to to let you know that we think that at some point what's happening with the standard curves is that close to the lower end there's a lot of variability. So we think the standards are not doing a good enough job as is in terms of quantifying the amount close to the lower end. One way to get around that, and that was used in the flu studies, was to use, instead of a 7-point standard curve, to use a 3-point curve, starting from standard number 3, just to look specifically. The reason is, I need to go step one back, you accumulate error as you dilute down. So obviously the reason that you have S, in this case S6, S5, and S4 doing better is not because something magical. It's just because we have a frozen stock. We keep a frozen stock of the standards. We aliquot that, and we thaw one aliquot every essay and start the dilution. Obviously, as you dilute down with day variability, you do better on the upper ones just because you accumulate error as you dilute down. So obviously, you're going to have more error close to the lower end, meaning S1, S2, S3 are going to be more variable day to day. So one way to go around this is, instead of only freezing standard number 7, you can also freeze a stock of standard number 3. Start from that one, see if you improve your reproducibility over time. And I think in the last slide here, you can actually see, this is for Luminex polystyrene. You can actually see, with one exception, the green assay, but pretty much I think it's a little tighter in terms of the S1 and S2, much less variable compared to what we had before.
So are you, with the error that comes with the higher dilution, is it also that you're reaching the limit of detectable sensitivity in the assay?
Well, that's a separate issue, because
You're essentially
yes.
able to distinguish between dilutions because the assay is sensitive enough.
But if that were true, then you should be kind of maxing. It should be the same close to the lower end. We do see variabilities. So it means
a little of both, actually.
Yeah.
I mean, in some of these, there is a bit of compression at the low end again, and it's
Yeah.
the curves spread wider in the middle, and I suspect that is the case. that
Yes, and
those are just hitting the bottom then.
You have higher dilutions, you're within sort of the sweet spot.
that's true. And actually, we have a slide that looks at the CVs overall of all cytokines and you can see basically that towards the lower end, you're hitting the limit, and so there's not much variability there because you're already at.
I mean, you might be able to continue to use a 7-point curve, which is a little nicer than a 3-point if you can improve the detectable sensitivity at the lower end.
Yeah, the only thing that I would point out here is that we really think that it's not really necessary to use that standard curve, because what we would kind of promote is to use a relative quantification approach. Most people, what they're doing is comparing either wild type to mutated, or non-treated to treated, or different time points, at a 0 time point to later time points. So if you plan your assay well and include the right controls and your right group that you're going to compare to, you're doing kind of a relative assay, comparing MFIs between the groups, rather than trying to say, this is exactly 5 picograms per ml. The other part that I'm not addressing here is at the upper, that's the lower end that I've addressed here. On the upper end, there are some very abundant cytokines like ICAM in serum. ICAM, if you try to quantitate that using the standard curve, what you hit is above 5,000 picogram per ml for most of them. So it doesn't really allow you to appreciate what you've measured there. Now, while it's not linear in that range, so a dilution, that's another point. When you dilute a sample, it's not a mathematical equation. It will not, if you dilute twofold, it may be twofold less, but it may not. It really depends on the cytokine, on the matrix, etc. So you can't assume that linearity leads to half the MFI, or double will if you just multiply by 2, if you diluted your sample twofold, you can multiply by 2. It's incorrect. It's correct mathematically. It's not really correct biologically. That's one thing. The other thing is that if you have a valid measurement, and you can see differences in that ICAM towards the upper end of the curve between your samples, and you're hitting the point where you say it's above 5,000 picograms, you're not saying much. If you show a relative median fluorescence comparison, you say, okay, I don't know if it's exactly 5,000 or 10,000, but I know that I have a measurement, and I can see a difference between one group and another. And that's where you're gaining, double gaining. Mm-hmm.
How comparable are the measures of NFI if we do it at a different time? Let's say we do samples for patients year 1 and with year 2. How compare? Could we use this to compare? It's not recommended.
Again, just repeat the question again.
Let's say we have patients that we're following several years, and let's say we do the cytokine assay at year 1 and cytokine assay at year 2. How could we make sure that the data is comparable on the NFI?
That's a very good one. That's a very good one. So I did not mention, and we can go into that more in detail in the demo. So we keep a stock of a certain 51-plex, and obviously we stock up quite a bit on that, but every year, about every year, we change a batch. Every time we change a batch, there's going to be some differences in the performance of that batch. So we're coming up with tools, and that's going to be presented in the later talks, in the bioinformatic talks. We're coming up with tools to allow you to compare data that was produced on one batch of 51-plex to other batches of 51-plex. And that's our best way to offer you reproducibility in addition to the control. One thing that I really recommend people to do in order to avoid as much possible switching batches is actually to plan their assays so that they let us know if they have 200 samples that they want to run. They want to run 50 now and 100 later. If they let us know, we can save some materials for them and then run them at a later time. The kits are good for approximately 2 years. Some even, we've tested some for 3 years, and they maintain their performance. So that's not an issue. Just to summarize some that we've been talking about, I think we can detect them, cytokines in picogram range. Our control sample offers us a way to follow reproducibility over time. We see that it's much more reproducible looking at the raw signal, meaning median fluorescence intensity versus the picogram per mil. However, you do get the picogram per mil data. We actually recommend to report the relative quantitation of the values in your samples. Standards that spiked in serum are detected at lower level. And again, I cannot emphasize more. Please aliquot upfront your samples. Every cycle of freeze thaw will really reduce your cytokine levels to 50, sometimes 80%, sometimes not detectable. So you can't assume that one freeze thaw is not going to do any harm. You have to try and plan your assay so that you can aliquot upfront and use aliquots as needed. I'm not going to go there. And this is just to reinforce that hopefully Sanchita's talk will elaborate more about normalization while preserving the rainbow of variability in your cytokines. And acknowledgements, obviously, to all HIM seers, Holden, Iris and Cindy, who did the work, Swati, which I'll present more of what she did. She helped with the quality control set up in the Luminex assay in the recent year, and all other members mentioned here. Thank you.
So, given if you only wanted to look at, half a dozen cytokines or something, what would be why would you choose MSD over Luminex or vice versa?
There are a number of reasons. First of all, if you're on a fishing expedition, many people prefer to go for the 51-plex because it offers them a better chance of detecting something that's going to be responding to what they're looking for. or varying in the sample that they're looking for. If you're not about to go for a fishing expedition and you want to target specific cytokines, you can go different ways. You can either come up with your own plex. We don't promote that, but we do allow that from time to time. So you can come up with your own plex if you want to purchase that and ask us to run that from time to time. Obviously, if it's going to be a very large study, it'll be a different consideration. MSD, for that matter, is our workhorse in terms of custom development, assay development, for other purposes as well. If you want to look at something that's not available and you want to develop an assay, that's a way to go, either with our help or your own work. Again, to some extent, the MSD for certain cytokines, really what it comes down to the differences between the antibody pairs used by the different companies. And we work a lot with for the 51-plex, we work a lot with the company in order to ensure that our detection is above the background level. So, yes, we can. Exactly. Exactly. So our custom one was very helpful in that sense, so that we could ensure that our background level is about, that our first standard is about twofold above background level. It's not accomplished for all cytokines, but for most of them. That's one thing that we do. For MSD, it's a given. They make their own pre-patent arrays. They do their own quality control. They have somewhat better linearity towards the lower end of the curve. So I think close to the limit of detection in the picogram range, the low picogram range, they may be doing better for some cytokines. It really depends on what you want to get and how abundant your cytokine is.
There's one issue, since you say abundant, because we've run into this with MSD.
Yeah.
You can get a bleed-through of signal if you have one really abundant analyte pattern in the same well with other low abundance analytes.
Is that just because those detection spots are too close together?
No, I think it's just light saturation.
Yes.
You know, it's like pointing a camera at the sun. You can't see anything dim around it.
And they're doing some corrections in there. So when it reads the spots, it automatically corrects based on the fluorescence. If you have one very high one, it may adjust all the rest of them there, even if they're very low. So it may be inaccurate to that extent. You have to have prior knowledge if possible. But if you don't, then